Hello dear children, welcome back. I am Dr. Jisha Joe. In this session, we shall study the structure of prokaryotic cell. In the previous video, we have seen what are prokaryotic cells. What are prokaryotic cells? Cells that do not have a well-defined nucleus. Well-defined nucleus is absent in prokaryotic cells. So the prokaryotes include bacteria, blue-green algae or cyanobacteria, mycoplasma, and PPLO, pleuropneumonia-like organisms. So, prokaryotes include bacteria, cyanobacteria. Cyanobacteria are the blue-green algae, mycoplasma and PPLO, which are the pleuropneumonia-like organisms. Now, prokaryotic cells or prokaryotes are small in size and they multiply at a faster rate than the eukaryotic cells. And they have a variety of shape and size. So prokaryotes include bacteria, cyanobacteria, mycoplasma, PPLO or pleuropneumonia-like organisms. Pleuropneumonia-like organisms, and they vary in their shape and size, and they are small and multiply at a faster rate when compared to the eukaryotic cells. Now, based on shape, bacteria are of different types. First one, coccus. Coccus means spherical. The shape of the bacteria is spherical. Example is Streptococcus pneumoniae. Coccus, the shape of the bacterium is spherical. Example is Streptococcus pneumoniae. Second one, Bacillus. Rod-shaped bacterium. Example, Bacillus anthracis. Bacillus anthracis, they are rod-shaped bacterium. The third type is Vibrio. Comma-shaped. Example is Vibrio cholerae. Vibrio cholerae. And the fourth one is Spirillum. Which has the shape of it's like a spiral. Example is Spirillum volitans. So bacteria are of different shapes. Based on shapes, bacteria are of different types. Coccus, they may be spherical in shape. Example is Streptococcus pneumoniae. Number 2, Bacillus, they are rod-shaped. Example, Bacillus anthracis. Third type is Vibrio, comma-shaped, Vibrio cholerae. And Spirillum, spiral-shaped. Example, Spirillum volitans. So, we have seen bacteria differ in their shape. And even though they have different shapes and they occupy or they live in various types of habitats, the regular organization, the body organization or the cellular organization is the same in all prokaryotes. That is what we are going to see now. we will have a general look at the prokaryotic cell. So any prokaryotic cell, if we take, will have an outer membrane which is called as the plasma membrane, and it will have an outer layer outside the plasma membrane which is called as the cell wall. Cell wall is absent in mycoplasma. So there will be an outer wall in all prokaryotic cells. An exception is mycoplasma. Mycoplasma do not have a cell wall. Do not have a cell wall. Then inner to the cell wall is the cell membrane or the plasma membrane, which is a semi-permeable membrane, and it is present in all the prokaryotic cells. And the fluid matrix that is present inner to the cell membrane is called as the cytoplasm. Cytoplasm, that is the fluid matrix that is present in all prokaryotic cells. Now, coming to the genomic DNA or the DNA in prokaryotes, we have already seen there is no well-defined nucleus. Well-defined nucleus is absent in prokaryotes. So, DNA or the genomic DNA which contain the genes, the DNA that contain the genes are located in the cytoplasm. And in addition to the genomic DNA, there is another circular DNA in bacterium which are called as the plasmids. Plasmids are used as vectors in genetic engineering or recombinant DNA technology. And so that is about the genetic material, the genomic DNA. It is present in the cytoplasm. No well-defined nucleus is present, so it is present in the cytoplasm. In addition to the genomic DNA, there is another circular DNA which is called as the plasmid DNA, and we have seen plasmid DNA is used as vectors in genetic engineering or biotechnology. And next is, so that is about genomic DNA. Now this plasmid DNA gives special phenotypic characters to bacterium. For example, there are plasmids which give antibiotic resistance to certain bacterium. So they are, these plasmids are actually single circular DNA molecules that give specific phenotypic characters to the bacterial cells. Then in all the prokaryotic cells, if you look, there are no membrane-bound organelles. Membrane-bound organelles are absent. Membrane-bound organelles are absent in prokaryotes. But ribosomes are present. Ribosomes are non-membrane bound organelles. Ribosomes are present in prokaryotes, and there are special infoldings of the cell membrane into the cytoplasm which are called as the mesosomes. Mesosomes. So this is I hope you have got a general idea about the structure of prokaryotic cell. Every prokaryotic cell will have an outer membrane which is the cell membrane. Then it will also have an outer wall which is called as the cell wall. An exception to this is mycoplasma. Mycoplasma do not have a cell wall. Then the fluid matrix is called as the cytoplasm. Membrane bound organelles are absent in prokaryotes. The DNA is not present within a nucleus. It is seen inside the cytoplasm. That is the genomic DNA, which is the DNA that contain all the genes, is a single circular DNA that is present in the cytoplasm. In addition to this genomic DNA, there is another single circular DNA which is called as the plasmid DNA, which gives special phenotypic features or characters to the bacterial cells. Like for example, antibiotic resistance is given to bacterial cells by plasmids present in them. And then even membrane bound organelles are absent, but ribosomes, which is a non-membrane bound organelle, is present in prokaryotic cells. And also there are infoldings of the cell membrane, which we call as the mesosomes. Now we shall have a detailed study about the structure of a prokaryotic cell. Now we shall study the structure of a bacterial cell, which is a prokaryotic cell. So most bacterial cells will have a complex cell envelope. An outer cell envelope will be present in most of the bacterial cells. This envelope is a three-layered membrane, or three-layered structure, which is tightly bound, and that is a three-layered structure, and the three layers are tightly bound to each other. The outermost layer is called as the glycocalyx. The middle layer is the cell wall, and the innermost layer is the cell membrane. So the three membranes together form the cell envelope. So most of the bacteria have the cell envelope. The cell envelope consists of three tightly bound layers. The outer layer is called as the glycocalyx, middle layer is the cell wall, and the innermost layer is the cell membrane. Now, in some bacteria, this glycocalyx layer is a loose sheath and is made up of polysaccharide. When it is a loose sheath made of polysaccharide, it is called as a slime layer. Slime layer. Whereas in some other bacteria, you can see the glycocalyx is thick and tough. Thick and tough. Tough layer, and it is formed of both polysaccharides and polypeptides. Then this layer is called as a capsule. Capsule. Okay, so that is glycocalyx is the outermost layer of the cell envelope in bacterium, and the glycocalyx, if it is a loose sheath which is made of polysaccharide, then it is called as the slime layer. Whereas in some other bacteria, when the glycocalyx is a thick and tough layer which is formed of polypeptides and polysaccharides, then it is called as a capsule. Now, the next layer is the cell wall. Cell wall in bacterium is formed of peptidoglycan. Peptidoglycan. Cell wall is formed of peptidoglycan. What are the functions of cell wall? Cell wall gives shape to the cell. It gives shape to the cell. It gives support, structural support to the cell. Support to the cell. And it prevents the cell from bursting. So these are the functions of the cell wall. Cell wall gives shape to the cell, gives structural support to the cell, and it prevents the bacterial cell from bursting. So we have seen the cell wall is made of peptidoglycan. The cell wall is tough and rigid, and the functions of the cell wall also we have seen. Now, the innermost layer of the cell envelope is the cell membrane. Cell membrane, like any other cell membrane or the cell membrane of the eukaryotic cells, here also it is a semi-permeable membrane. We shall study more about it after a few minutes. So once again, cell envelope, that is the outermost layer or the outer covering of the bacterial cell, which is made up of three layers, or it is a three-membraned structure or three-layered structure. The three layers are tightly packed. The outermost layer is the glycocalyx. If the glycocalyx is a loose sheath formed of polysaccharide, it is called as the slime layer. If it is a thick and tough layer formed of polysaccharides and polypeptides, then it is called called as a capsule. Coming to the cell wall in bacteria are formed of peptidoglycan, and it is a tough and rigid layer, and the functions of the cell wall are it gives shape to the cell, it gives structural support to the cell, and it prevents the cell from bursting. Now, based on the differences in the cell envelope and the manner in which they take up the stain, bacterial cells are of two types: gram-positive bacteria and gram-negative bacteria. As you can see here, I have written down the features of gram-positive bacteria. They take up the stain. When stained, using the gram stain, they take up the stain easily. That is why they are called as gram-positive bacteria. Whereas in the case of gram-negative bacteria, do not take up the stain. Okay, they do not take up the stain. They do not get stained. Then coming to the cell wall, in the case of gram-positive bacteria, the cell wall is thick, whereas in gram-negative bacteria, the cell wall is thin cell wall. In gram-positive bacteria, the lipid content is low, whereas in the case of gram-negative, the lipid content is high. Gram-positive bacteria are more resistant to physical disruption, whereas gram-negative bacteria are less resistant to physical disruption. And example of gram-positive bacteria is lactobacillus. Example of gram-negative bacterium is E. coli, Escherichia coli. So these are the differences between gram-positive and gram-negative bacteria. This is based on the difference in the cell envelope and the difference in the manner in which they take up the stain. Bacterial cells are classified into two: gram-positive and gram-negative. So gram-positive bacteria take up the stain easily, whereas gram-negative bacteria do not take up the stain. They do not get stained. Then cell wall is thick in gram-positive bacteria, whereas cell wall is thin in gram-negative bacteria. The lipid content is low in gram-positive bacteria, whereas the lipid content is high in gram-negative bacteria. Gram-positive bacteria are more resistant to physical disruption, whereas the gram-negative bacteria are less resistant to physical disruption. Example of gram-positive bacteria is lactobacillus. Example of gram-negative bacterium is E. coli. Now moving on to the innermost layer of the cell envelope, which is it? It is the plasma membrane. It is a semi-permeable membrane and it is lipoproteinaceous in nature. That is, it is formed of lipids and proteins. And in prokaryotes, in bacterial cells, if you look, we are studying the structure of bacterial cells. In bacterial cells, if you look, the cell membrane or the plasma membrane has several infoldings which are called as mesosomes. So I am just drawing the structure of a bacterial cell. So we have the cell, okay. It is formed of the outermost layer, which is the glycocalyx. We have studied in detail about the glycocalyx layer. When is it called as the slime layer? When is it called as the capsule? Then we have discussed about the cell wall. Then the innermost layer is the cell membrane. The cell membrane has several infoldings like that. Okay. So that is the innermost cell membrane. So once again, outermost glycocalyx. Okay. Then the middle layer is the cell wall. Innermost layer is the plasma membrane or the cell membrane, and it has several infoldings which are called as the mesosomes. Mesosomes are the infoldings of the plasma membrane into the cytoplasm. Now we shall have a look at the functions of the mesosomes. Mesosomes help in cell wall formation. They are concerned with the formation of cell wall. Second one, they are concerned with DNA replication. Mesosomes are concerned with DNA replication. They are the reservoir of respiratory enzymes, so they help in respiration. They help in secretion, helps in secretory processes. Secretory processes. And mesosomes help increase the surface area of the plasma membrane, and they have they are, as I told you, they are a rich source of various types of enzymes. Increases surface area of plasma membrane and rich reservoir of enzymes. So these are the important facts to be remembered about the mesosomes. So mesosomes, we know they are the infoldings of the plasma membrane into the cytoplasm, and they are concerned with the cell wall formation in bacteria. They are involved in DNA replication, help in respiration since they have a lot of enzymes that are concerned with respiration. They help in the secretory process, also increase the surface area of the plasma membrane and has a rich enzymatic content in them. So that is with regard to the mesosomes. In cyanobacteria, we have seen cyanobacteria are blue-green algae. They can perform the function of photosynthesis. So in cyanobacteria, certain membranous extensions are there into the cytoplasm, which are called as chromatophores. Okay, so chromatophores are present in cyanobacteria, chromatophores. They are also extensions of the plasma membrane into the cytoplasm, but they contain the pigments that are necessary for photosynthesis. So chromatophores are present in cyanobacteria. So we have till now we have seen the cell envelope, which is formed of glycocalyx, cell wall and the plasma membrane. We have discussed about the glycocalyx, cell wall and the plasma membrane, and we have seen that on the plasma membrane there are infoldings present which are called as the mesosomes. We have also discussed about the functions of the mesosomes. Then the fluid matrix that is present inside, what is it called? The cytoplasm. That is the cytoplasm there, cytoplasm. Now bacteria are of two types based on their ability to move. Those bacterial cells that can move are called motile forms, whereas the ones that do not move are called as the non-motile forms. Now in motile forms there is a structure which is called as the flagella. Flagella is plural, flagellum is singular. These flagella are filamentous extensions from the cell wall in the form of proteins. They are formed of proteins called flagellin. So they are extensions of the flagellin. They are extensions of the cell wall, filamentous extensions of the cell wall, and they are formed of a protein which is called as flagellin. Flagella are present only in motile forms because they are concerned with movement of the cells. And if you look at the structure of flagellum, each flagellum will have three parts. Okay, three parts are there. There is a basal part that is seen enclosed within the cell envelope. That is the basal part there. Then there is a small hook-like region. And then there is the long filamentous structure which is called as the that is the filament. So three parts, that is the basal body, then there is the hook, and that is the long filament. So these are the three parts of the flagellum. So which are the three parts? The flagella has a basal body. This basal body is seen enclosed within the cell envelope. We know what is the cell envelope. So basal body is enclosed within the cell envelope. Then the hook region, it is a slightly curved structure which is seen close to the cell surface, and then there is the long filament. So these are the three parts of the flagella. Okay, and flagella are seen in motile forms of bacteria. They are filamentous extensions from the cell wall. They are formed of a protein called flagellin, and each flagellum consists of three parts: the basal body, which is enclosed within the cell envelope. Then there is the hook region that is seen close to the cell, and the innermost region, which is the outermost long filamentous structure. So these are the three parts of the flagella. Now that is the long flagellum that is seen there. Okay, that is the long flagellum. Now other extensions from the surfaces of the bacterial cell are pili and fimbriae. So these are also surface structures that extend from the bacterial cell, but remember, these are not involved in motility. They are not concerned with motility, so these structures are present in both motile and non-motile forms of bacterium. So pili, they are elongated tubular structures, and they are made of a protein called pilin, the protein that is involved in the formation of pilin. So they are elongated tubular structures that arise from the cell surface, and certain pili, which are called as the sex pili, are involved in the transfer of genetic material from one bacterium to another during bacterial conjugation. So during conjugation, where the genetic material from one bacterium is transferred to another bacterium. That process is called conjugation. During the process, the two bacterial cells have to remain close together. So this is done with the help of sex pili. Okay, so that is with regard to pili. Pili are formed of proteins called pilin. They are present in both motile and non-motile forms. They are not concerned with motility. Certain pili are called as sex pili, which are involved in the transfer of genetic material from one bacterium to another during bacterial conjugation. Now coming to the second structure, that is the fimbriae. Okay, fimbriae are also not concerned with motility. Okay, so they are present in both motile and non-motile forms. They are small bristle-like structures. Present on the surface of the bacterial cell, and they are concerned with or they help the bacterial cell to attach itself to a substratum or to the host cell. So the fimbriae helps in attachment of the bacterium to the surface or to a host cell. So that is with regard to pili and fimbriae. Remember, they are not concerned with motility, so they are present in both motile and non-motile forms. Pili are elongated tubular structures extending from the bacterial cell surface. They are formed of a protein called pilin. Sex pili are involved in the transfer of genetic material during bacterial conjugation. Whereas fimbriae, they are bristle-like structures arising from the cell surface, and they are concerned with attaching the bacterial cells onto a surface or onto the host cell. So that is with regard to pili and fimbriae. Before moving on to the next, I shall just mark the flagella. That is the flagellum there. Then extensions which are called as the pili and the fimbriae. Okay, so tubular extensions are the pilus or the pili. These are the pili. Then fimbriae, bristle-like structures. Bristle-like structures arising from the surface of the cell, and we have studied the functions of each. Next, we are moving on to the genetic material. We have seen it is not enclosed within the nucleus. It is present in the cytoplasm in the form of single circular DNA. That is the genetic material there. You can see the genetic material. And since it is not enclosed within the nucleus, it is called as the nucleoid. Okay, nucleoid. The genetic material in bacterium is called as the nucleoid, and it is in the form of a single circular DNA which contain all the genes, and this is present inside the cytoplasm. Okay, so and the nuclear material of or the genetic material of bacterium is called as a nucleoid. Now, in addition to the nucleoid, an extra chromosomal genetic material is present. Extra chromosomal hereditary material is present, which is a small circular DNA, which is called as the plasmid. As we have already seen, plasmid confers certain unique phenotypic characters to the bacterial cells, such as antibiotic resistance, and plasmids are used in genetic engineering or in DNA recombinant DNA technology. Plasmids are used as vectors in genetic engineering and recombinant DNA technology. So that is with regard to the genetic material. So the genetic material, or the since in bacterium the genetic material is not present enclosed within a well-defined nucleus, the genetic material in bacterium is called as a nucleoid. In addition to the nucleoid, there are plasmid DNA also present. Now moving on to the next structures, that is the ribosomes. Ribosomes are non-membrane bound organelles. They may be seen present in the cytoplasm or may be seen associated with the plasma membrane. The ribosomes present in bacterial or prokaryotic cells is the 70S ribosomes, which is formed of two subunits. The two subunits are 50S and 30S. So the ribosomes in prokaryotic cells is 70S ribosomes, and the two subunits of prokaryotic ribosomes are 50S and 30S. Ribosomes in the prokaryotic cells may be seen associated with the plasma membrane or may be seen in the cytoplasm. And the function of the ribosomes are the sites of protein synthesis. And in prokaryotic cells, sometimes several ribosomes may be seen attached to an mRNA. To a single mRNA, you can see several ribosomes attached. And this structure, that is a single mRNA. So function of ribosomes is to synthesize proteins. So on the mRNA, you can see several ribosomes attached like that. Okay, several ribosomes may be seen attached, and each synthesizing a protein. Okay, so such a long chain in which several ribosomes may be seen attached to a single mRNA. This is protein. These are proteins that are being synthesized from the mRNA. Okay, so in prokaryotes, you can see several ribosomes attached to a single mRNA to form a long chain, and this is called as a polyribosome or the polysome. Polyribosome or polysome. So what are the ribosomes present in prokaryotic cells? They are the 70S ribosomes, which are the subunits 50S and 30S. And in prokaryotes, polyribosomes or polysomes are seen. What are they? Several ribosomes may be seen attached to the mRNA chain. And this is called as the polyribosome or polysomes. So that is with regard to the ribosomes. So ribosomes may be seen. So these are the ribosomes. They may be seen in the cytoplasm or may be seen attached, associated with the plasma membrane. Now moving on to the last structures that you can see in the prokaryotic cells, they are the inclusion bodies. What are inclusion bodies? Inclusion bodies. So reserve materials in the prokaryotic cells are stored within the inclusion bodies. The reserve materials are stored in inclusion bodies. They are not bounded by any membranes, but they are present in the cytoplasm. They are found freely in the cytoplasm. They are the inclusion bodies. They are or they store the reserve materials. Examples of inclusion bodies are the phosphate granules, cyanophysian granules, glycogen granules, all these are examples of inclusion bodies. So inclusion bodies store reserve materials in prokaryotic cells. They are not bounded by any membranes. They are seen freely lying inside the cytoplasm. Examples of inclusion bodies are the phosphate granules, glycogen granules, cyanophysian granules, etc. Then in photosynthetic bacteria, blue-green bacteria, purple and green photosynthetic bacteria, we can also see gas vacuoles present inside the cytoplasm. So children, in this video we have studied the detailed structure of the prokaryotic cell, and I hope this session is clear to you. If you have any doubts or suggestions, please give your doubts and suggestions in the comment section. Thank you.